how many of you already read the paper that you have to discuss next week? More than one person? No, good. Because I just remember that I sent you another, not the paper I wanted to discuss. So you can read it, but I'm going to send you another one. And I think what we'll do is instead of doing it on Tuesday, because Yoram is cannot do the presentation on Tuesday, so we'll do Tuesday another class, and then Thursday we'll do the paper, so you have time to read. So I'll send you today the paper, and then you'll read it to next Thursday.
Also our papers?
Yes, yes. Yeah. So we'll do the paper session next Thursday, and I'm going to send the class another paper to read instead of the one that I sent you. Okay, and on Tuesday we'll have another class on a different topic. Okay. So we continue where we stopped last time. I think you probably don't see anything, right? Why is it not working? Okay. So we talked about LTP, and we mentioned that that LTP is NMDA receptor dependent. And then the next question that was actually it's a kind of maybe we okay. So let's come back to this question in a second. And also this one. So the next question that people started asking themselves is whether it's a presynaptic or postsynaptic. Where does the change in the synapse takes place? It is the button that is presynaptic to the synapse, or the postsynaptic site. And there are evidence. I mean, this was a big, one of the hottest topic. And people kind of, it's like the world was divided, and everybody had a side, and there were wars and everything. I don't know exactly where it stands today, but it's more like everybody knows that it happens on both sides. But so I just want to give you two kind of examples how people try to attack this problem. So one thing that they noticed is that on the people that, on the side of the people that said that it was postsynaptic, they would kind of show examples where you would do LTP. If you look at the EPSP amplitude, you see that the EPSP amplitude has changed over time, like we saw before. But if you look at what they called paired pulse ratio, which means instead of giving like one spike presynaptic and look at the EPSP, you give one and another one just after it, and you measure basically two EPSPs, one for the first spike, one for the second spike, and you look at the ratio between the size of the EPSPs. So the ratio is the idea was that when you do that, the second EPSP would not be the same size of the first one because the presynaptic release site will not release the same amount of neurotransmitter in the second pulse, and this is usually an indication of presynaptic process. And the paired pulse ratio did not change with time. So this meaning that, there's no more glutamate in the vesicle, or the probability of release is not changed. In the other direction, people showed examples where actually you have exactly the opposite result. So if you look on the right here, you see that the failure rate was, so what is a failure rate? Is like you give a pulse and you look for an EPSP, and from time to time you don't see an EPSP. Okay, and this is because the synapse is failing to release transmitter. You know about synaptic failures, right? It's not like the first time you heard about it, or you do. You don't? All right. Oh my God. Okay. This is a whole new class. Okay, so anyone heard about Bernard Katz and Milady? So there's a Nobel Prize laureate. He actually worked already with Hodgkin and Huxley. And he was actually the first one to study synapses in really great detail. And one of the first things that he noticed, uh, he worked on the neuromuscular junction, so between the nerve and the muscle. Okay, and one of the first things that they showed is this quantum release. So when you stimulate the neuron, the response is quantized. So you get like different size of responses, but they are in jumps. Okay, so one of the things that they noticed right at the beginning is that many times when you give a stimulus, you don't get any response. So synapses are stochastic in nature in terms of releasing the synapse. It's kind of a stochastic process, and many times you just don't get any release. So I'm not going to go into that. I'll prepare a different, proper presentation of that. I will give you a paper presentation of Katz and Milady. But what you can see here is that when they do the presynaptic stimulation, they have a release probability of 0.4. Okay, so 60% of the time, the synapse doesn't release glutamate, and you don't get a response in the postsynaptic cell. But after you do LTP protocol, this drops to 0.2. Okay, so in a way, what they suggest here is that the change that you get in the postsynaptic side, in the LTP, is not a change in the size of the EPSP because there are more receptors, for example, but because you now release on average more, the chances to release is higher. Okay, so this means that you get 0.8 or 80% of the time you'll get release. Okay, so now if you'll do that for, say, with many synapses, the average EPSP will become larger because every time that you stimulate a bundle of axons, you'll get more spikes. You get more release. Yes.
How is that related to the NMDA receptor side? I mean, the fact that
that's a good question. Let's talk about it. This is I think in the days before NMDA even.
What is LTP protocol?
LTP protocol, it means like this high frequency stimulation. Okay, and at the same time, well, okay, so so I'm not I don't want to kind of go into all the details of this process, but what I want to show you is that first of all, it was a big question whether it's postsynaptic or presynaptic, and in some ways we know that today that it happens on both sides, but the details are still not kind of figured out completely. One of the big questions is, for example, that we know that it's NMDA dependent, okay? But as Nir asked, if it's NMDA dependent and NMDA is on the postsynaptic side, how is that going to work on the presynaptic side? Okay. So first of all, there are also NMDA receptors on the presynaptic side. But this wouldn't explain everything, because in some cases, for example, if you do this knockout experiment, you can show that you need the postsynaptic NMDA receptor rather than. But okay, so there is NMDA, and what happened? Why is this NMDA important at all? So the idea is, without actually going into all this kind of crazy stuff that's going on here, is that when you have NMDA is permeable for calcium. So once you open NMDA channels, calcium gets in. So it's not only that the voltage change, it's calcium getting in. And calcium is a main signal carrier in neurons. So when calcium gets in, it can interact with IP3 receptor, it can act with G protein coupled receptors, with all kinds of things. activate all kind of biochemical pathways, and the idea is that this is triggering the whole process. So all these things happens on the postsynaptic side, but still we have a problem how that happens on the presynaptic side. So one option was this. You see here this thing called NO. So NO is a molecule, the small molecules that can diffuse through membranes. So one of the ideas was that when calcium gets in, it activates all kind of biochemical pathways, and eventually NO goes out of the synapse, diffuses out, and acts as a messenger, as a retrograde messenger to the presynaptic cell. This happens with NO, and today we know that there are other molecules like that. For example, endocannabinoid, which is the same, basically the endogenous form of cannabis, okay, of the active ingredients of cannabis. Endocannabinoid is also retrograde messenger that goes back between the presynaptic and the postsynaptic side, and there are actually receptor for endocannabinoids on the presynaptic side in some type of synapses. And there are also NMDA receptors on the presynaptic side, and and one of the hottest topics today that people are starting to talk about but still don't know, it's really it's still not figured out completely, is that these synapses, it's not like these two things are just floating around, the presynaptic side and the postsynaptic side. There's molecules that are coming out of the postsynaptic cell and out of the presynaptic cell. One of them is called neuroligin. Okay, so neuroligin is coming out from both, and it has two forms, and when they meet, they hold each other, and they're actually making a construct that holds the synapse together. Okay, and so synapses like that has postsynaptic, they have something inside the membrane of this cell, and they have something inside the membrane of that cell. And in some of the ideas is, and these are not the only molecules that coming between the two sides. So one of the ideas is that through these molecules there are signaling that are going between the two sides. Neuroligin. So you have a membrane that's coming from the axon of the presynaptic cell, and then a synapse that comes from the postsynaptic cell, a spine. And it's not that they're just close to each other. They're actually held together. It's called adhesion molecules. It's like glue that glues them together. Now, they're not really stuck to each other. They hold at a distance. But the distance is extremely small. It's 200 angstrom. It's really small. It's like smaller than a vesicle. Okay, so the vesicle, when it opens, it cannot actually, a vesicle cannot be stuck between the two sides. It's really, really close. Okay, so it's actually held together by these molecules that are holding it.
Which are holding both sides of the...
Yes. So one of them is sending, one of them is expressing a protein that comes from here, and one of them is expressing a protein that comes out of here. And when they meet, they're actually stuck to each other, and they hold, and there are many of them, and they hold the synapse. And in a way, this is also helps to encapsulate the synapse, so things doesn't diffuse too much outside and everything. Okay? So the whole, in a way, the whole issue of synaptic physiology is a huge section of neuroscience. I will try to do a lecture on that on the end of the course, but because it's extremely important in terms of like signaling between neurons, all this plasticity that's going on. So there are huge. It's a huge part of neuroscience is understanding the details of all these processes, and we'll touch upon just really today on few of them. I just want to mention few things.
It's like a closed loop.
In what sense?
Because they interact both sides. It's a closed loop, so they talk to each other.
They talk to each other, but the idea is that you start with some kind of electrical signal, it becomes biochemical. If it kind of triggers this calcium entry into the postsynaptic site, then the whole thing, kind of change both on both sides. Okay, so this is one type of plasticity. Okay, I'm going to go to show you now another type, and then maybe I'll go back to these slides and I'll mention even a third type. So somewhere in the 90s, I think it was 97 or something around this time, people started to record from pairs of neurons. Okay, and they noticed there were one of them, the first one I think was not the first one, but one of the major players was Henry Margrams, that you probably heard about from this Blue Brain project. This is from another group by Mu-Ming Pu and B. He was his postdoc. What they did, instead of doing these like trains of presynaptic, very high frequency trains, they actually played the game where they stimulate one neuron, one cell, and then at a later time stimulate the second cell. Okay, and what they actually were interested is if they do that enough times for a different, for a certain delay between the presynaptic and the postsynaptic, how is that going to change the synapse between the two? Okay. So what you see here is a graph. So what you see here is the following. You see, okay. So this is what they called a positive delta T, meaning that the postsynaptic cell. So this would be where the presynaptic cell is being activated, and this is when the postsynaptic cell is being activated after a delay. So if the delay is of about 20 milliseconds, you get this amount of potentiation. So the synapse starts at 0. 0 meaning, every time you give a pulse, you get an EPSP, and the EPSP has a certain size. So we call this this is baseline. Now we stimulate the presynaptic cell, and then the postsynaptic cell 20 milliseconds afterwards. Repeat that 100 times. Okay? And then just stimulate the presynaptic cell without the postsynaptic cell, looking at the EPSP, and we see that the EPSP becoming larger. Okay, larger by about, 20%. Yes.
Current though, right? It's not EPSP.
Yeah, okay, so this is EPSP. So it's done in voltage clamp. When you measure, you're doing it in voltage clamp. If you do it 10 millisecond delay, then it's much higher. Okay? If you wait for a long time, like 80 milliseconds, you don't change the synapse. Okay? But now you do the experiment in the reverse direction. So you first do the postsynaptic cell, and then a little bit afterwards the presynaptic cell. Okay, so this is negative delta T. And on a negative delta T, what you see is that the synapse actually depressed. Okay, so if you constantly stimulate the postsynaptic cell before the presynaptic cell, the synapse become weaker. Okay? But if you do it like 100 milliseconds before, it doesn't matter. Okay, so there is a window which is around 40, 50 milliseconds, in which so the whole window is about 100 milliseconds, and in this window, if you have coincidence of these two neurons, the synapse is going to change. If the coincidence in always the same direction, then it's going to change in a consistent manner, okay, according to this this curve, and this was termed later spike timing dependent plasticity, because the plasticity is dependent on which is coming first, and this in a way give you just the feeling of this Hebbian rule, right? So if two neurons are firing together, the second one is firing after. So it's not just Hebbian; it's kind of causal rule. So if the postsynaptic cell firing after the presynaptic cell, it meaning like the synapse helped this cell to fire, then I'm going to make this synapse stronger. Okay? But if it wasn't, if this synapse is meaningless, it's like tend to activate in the wrong direction. So it's kind of activate after I already made a spike. So I'm a postsynaptic cell, and I have a synapse, and I'm spiking, and then 10 milliseconds after I get this input. So this input seems to be irrelevant for me to spike, because always coming after I already spiked, so I don't need it, right? So I can actually chuck it. I make it weaker and weaker, and I get rid of it. The interesting part is what happens really on the 0 millisecond, right? So it's kind of this like a discontinuity, which is really strong. You go from depressing the synapse very, very strongly to increase the synapse very, very strongly. As if the neuron really cares about synapses that comes right before it spikes. Okay, and it doesn't care about synapses that comes right after it spikes. Okay, these synapses are really, really not important in a way. Okay? So this started, again, a whole new field of research. People tried it in many, many neurons, and this is not the only consistent, this is not the only way these curves is seen. So there are neurons, for example, that have completely the reverse curve. Okay? So actually, if the synapse comes after you spike, then it becomes potentiated. And if it comes before you spike, it's actually being depressed. So it's kind of from theoretical point of view, people try to figure out why is that important. It's not obvious. But there are many, many computational studies on this curve. People try to do all kinds of simulations. So I'm just mentioning it here. Okay, questions?
So just to better understand the experiment, they gave both pulses, and then they waited, and then they gave another pulse and measured the.
Okay, let me draw the experiment if it's not clear. I'm sorry. Oh, sorry, I'm going to the wrong. Okay, so here's what the experiment looks like. You have two neurons. Okay, you have a patch here and a patch here. You call it. You call this one. You say this is a postsynaptic, and this is a presynaptic. And why? Because when you give a pulse here, and you record a spike, so a little bit afterwards, you'll see here an EPSP. Okay, so there's a synapse that comes from here to here. Okay. So this is a presynaptic neuron, that's a postsynaptic neuron. Is this too small for you to see, or you see? Okay. So now you do that, and you measure the amplitude of that thing. So actually you do that in voltage clamp, meaning that what you measure is something like that. It's a current, okay? And you measure the current, and you make a plot, and you repeat it, and you see something like that. Okay, so the amplitude is kind of more or less the same. Sometimes you don't get anything, sometimes you get, and when you get something similar. Okay. Now you start doing the following. You inject current here, and then a little bit afterwards. So say this is time 0, here and here. Okay, this is time 0. So at time 0, you give a pulse here, and your neuron is doing a spike again. Okay. Here, this is time 0. You wait 20 milliseconds, and then you give a pulse. So your neuron would do something like that. It will start the CPSP. Now we're in current clamp. Okay, we switch. The postsynaptic neuron is now in current clamp. We measure voltage. Okay, so a neuron will start the EPSP. Okay, and then sometime here you will make a spike. Okay. So this means this is a delta T of 20 milliseconds, 10 milliseconds, and then you repeat that. You repeat that 100 times. I don't remember the specific details. Doesn't matter. Okay. You do that for 100 times, then you go back to this experiment. Okay. Now you stimulate here, and what you see is that the synapse become larger. Okay. So what you will see here is like an increase in the EPSP. Okay. So this was one point on this graph. So if we plot this graph, okay, so this would be here, and you measure the difference between that and that in percentage, and you place a point on the graph. Okay, now you do it again with a different two different neurons now from scratch doing exactly the same thing. Uh, Only here now you change this, and now you give this stimulus before. So what you get is a spike, and then the EPSP. Right? You do that 100 times, and then again you come here, and then what happens is the synapse becomes smaller. Okay, so you come back and you make a point here. Okay, and after you repeat it for different times, for many neurons, you get this kind of curve.
So in a way, it's a bit strange that it's exactly at zero, right? If there's a time that it takes for
So
saturation.
the problem is that time zero is kind of it's a kind of a singularity point, because it's not always when you give the spike, when you give the stimulus, the spike doesn't actually start immediately when you give the stimulus. It sometimes had like, half a millisecond delay to here and there, and there's also conduction velocity between the two.
Yeah, that's
All right. So zero is kind of so there's a lot of variability. I mean, this is kind of there's a lot of variability, but you can also see it, when you actually look at the original graph. Um, and I guess what they did here is like just did it, so many repetition and average. So every point here is like an average probably of many neurons. So it looks clean, but the data itself is not that clean. Okay. So I said that I'll mention another type of plasticity, and this is what we call the homeostatic plasticity. So what people notice that, for example, if you take a culture, okay, you talked a little bit about cultured neuron in the method course. So you can actually take neurons where when the animal is very young, and then put it on a plate, and then put it in an incubator, and the neurons would grow, and you can actually hold them for like a month or 2 months, like that. Okay, and they're still alive. And they make synapses with each other, and they spike, and they're not like neurons in the brain, but they are neurons, and it's a kind of small neural network. So for example, you can take a synapse like a culture like that, and put TTX on the culture for 24 hours. So for 24 hours, no neuron in the culture can spike, because all of them have to be silent. You block all the sodium channels. Okay? So what you do is you record before you put the TTX. You record from a neuron, and you kind of look at the average size of the EPSP that it gets spontaneously. Okay, you do it for 5 neurons, say. Okay, then you put TTX for 24 hours. Then you take another neuron, and you record the size of the EPSPs. And what you see is that all the EPSPs become larger than they were before you do this blocking with TTX. Okay, as if the neurons feels that it doesn't get any input, it makes its synapses more sensitive. Okay. You can do the opposite. You can put like KCL, which makes all the neurons spike all the time, and then actually the synapses become smaller. But it's not like one synapse becomes smaller. It's all the population of synapses in this network becomes either higher or lower. So this is called homeostatic plasticity, because it's kind of normalizing all the synapses to a reasonable range, so they will be kind of effective. Okay? And so this was the first experiment that people did about it, and today there are evidence that actually there are some of the communication between neurons on the pre and postsynaptic side are trying also to maintain some homeostatic plasticity. So like on a single dendrite, you would find synapses that are similar in terms of like the amplitude. And they're kind of trying to match all kinds of parameters like synaptic release or EPSP size. So there are all kinds of complicated phenomena that go in here, but these experiments are extremely difficult because they are very long term, and you have to kind of watch a single synapse over a long time. Yes.
So you were talking about the change on the synapse on the previous slides as well. So the results don't show that, right? I mean, they show a change
You mean on this one? Sorry,
On the
this one?
yeah, on this one. So we can see in the results that the reason for that is a change on the synapse. The question is,
What do you mean?
I mean, it could be that the change is within, let's say, the postsynaptic cell soma or whatever.
But you can measure many parameters and kind of exclude that.
So are there any morphological evidence that
For yes.
that show that the change is indeed in the synapse in this kind of protocol, for example?
Um, I don't know if in this type of experiment, but in LTP there are certainly there are certainly experiments that shows that the button size become larger. Okay, or there are evidence that one of the things that people have showed is this AMPA receptors. Okay, could they have like different? They could actually be in such a position, such that the receptor side is in the outside of the membrane. So if glutamates come, if the channel opens, all right. But they could also be what's called internalized inside the membrane. So this part would be actually, so it shifts really small distance, but it's actually inside the membrane. So this channel is now not active, okay? Because if glutamate comes, it cannot bind to the channel. But if you phosphorylate this internal side of the protein, it goes out. Okay, so by a small manipulation, you can actually add AMPA receptors into the membrane, even without making the protein. Okay, the protein is there, it's just not active. You make it active. Okay, so there are all kinds of details like that that you can show that there is actually a physical change in the synapse.
Yeah, but this needs to be proven because even at the PTX experiment you talked about, so you see bigger responses.
And you can also correlate this one, you can correlate with size of synapses.
It can be from multiple resources within the cell somas after 1 day without spiking. I mean, it's not obvious.
The soma, that's what I'm trying to understand is what do you mean by the soma? What is the soma kind of has to do with it?
Cell body?
No, cell body, yes, but what is the cell body special about?
No, that's the place, I guess, where, I mean,
that you record.
The EPSPs are being integrated, or...
Okay, but they're integrated, but if you look at the individual EPSP, why would the soma make any difference to it?
It could be that the same current is going in from the synapse, but something different occurs inside such that the EPSP we record is bigger.
It's true. Yeah, we're going away out of this, from this topic. There are differences also. Everything in the cell can change. The amount of every ion channel can change, and the combination of ion channels, and where these ion channels on the membrane are, and many things can change, and they do change. Okay, but some do correlate with what you do, and some don't. All right, so it's like there are kind of, from an experimental point of view, there is a protocol to do that, and there is a protocol to do that, and they don't always come together. So you can somehow separate. You can change the synapse without changing the amount of ion channels, and you can change the ion channels without changing the synapse. And they both actually, when you think about it, there's like a role for both. Okay, because sometimes, like in the case of the homeostatic synaptic plasticity, you just want to make the neuron more sensitive. You don't need to change all the synapses. You can just make the neuron more sensitive. You can make, like, for example, more sodium ion channels, so the threshold become lower, and then you will make the spikes coming easier, right? So you can do that. So it's a global change, or you can change like individual synapses. In this manipulation that I told you about, when you put TTX on the synapse, it's like as if someone will put you in hospital for a day and keep you on an anesthesia for a whole day, but a very strong one that your brain is completely silent. So when you wake up, you might be a little bit kind of overexcited. Okay, it's kind of so it's such a strong manipulation that it's not surprising that both synapses and ion channels are changing. Okay, let's go on because we are really behind schedule. Okay, so I'm going to skip that. And I want to just highlight few of the really, really important questions that are coming out of this issue of synaptic plasticity. So um, uh, one of the things that I haven't shown you is that protein synthesis, oh, maybe, do I have it here? Didn't we talk about it? Oh, I probably just skipped it. Sorry. Okay, let's skip it. It doesn't matter. One of the things that you can do, which is a kind of, again, it's a little bit brutal thing, is you can stop protein synthesis. Okay, so you just kind of block all protein synthesis in the cell, and then you can see that LTP you don't see LTP. So you need to synthesize more protein in order to have LTP. Okay, at least in some cases. Okay, but protein synthesis is traditionally thought of being done in the nucleus. And you saw already that, like in pyramidal cells, you can have like synapses that are a millimeter away from the nucleus. So how does the nucleus actually knows that a synapse somewhere there has been changed? Okay, so this was a big mystery, and it's still not obvious. One of the things that are kind of known today is that there's a lot of local protein synthesis next to the synapses. Okay, so there's always mRNA for certain proteins that is shipped into every location in the cell that can have synapses. This mRNA will stay there, but it will be somehow inactive, and there are ribosomes there. So many of these processes can happen locally. But it's not obvious that this is the only thing that takes place. Okay? Similarly, you can ask yourself, how do the proteins knows how to get, where to get? So say you had an LTP protocol in a certain synapse, and there is a protein that is synthesized, and you need it in the synapse in order to do this LTP. How does the protein knows where to go? Okay, so there is a whole interesting topic called synaptic tagging. So the idea is that when you do when you have a LTP in a synapse, you have some tag, some biochemical marker that when the protein is shipped along the tree, it will actually, being held there, and it knows, this is where I have to go. This is a very interesting process, but we don't really understand it completely. The other thing that you can ask yourself is, okay, so we made an LTP in the synapse. Now we need, twice as much of AMPA receptor in the synapse than we had before. Okay, so let's phosphorylate some AMPA receptors, and now we have twice. But now these proteins, while the neurons, stays for your lifetime, it's the same neuron that you were born with, and maybe even the synapse is the same synapse that you were born with. The AMPA receptors are certainly not, the same AMPA receptors. They're being all the time being exchanged. Okay, so they last for few days, sometimes weeks, but they don't last. It's a protein, an active protein in a cell like that is kind of being recycled all the time. So it's being taken out of the membrane, being degraded, and then another one will come in. So you need some local thing or a global thing. Something needs to know how many AMPA receptors needs to be on every synapse. So this is a very complicated mechanism. Obviously, there's no logbook somewhere in the nucleus that says, this synapse needs that, this synapse needs that. So you need something which will maintain it, and it will maintain it on a very, very stabilized process. Otherwise, synapses will very, very quickly forget what they actually learned. And again, we talked about, the communication between the pre and post-synaptic cells. Okay, so just to give you an example on how big is this topic. This is a chapter about synaptic plasticity from the hippocampus book that I showed you. Okay? So the chapter ends on page 444, That's where the reference starts. And 30 pages later, it ends. So it's 30 pages of references. Okay, and this is not like up-to-date book. So this is like really crazy. It's really difficult to maintain, knowledge about all this process. Uh, but even though this has been studied for 40 years, uh, the big question is still out there. Actually, some people claim to just solve it, and we might actually do this paper, but I haven't actually talked about memories. That's what we're going to do now. But uh, the whole motivation, in a way, to study synaptic plasticity, except that it's like, it can supply work for many, many scientists, is that from early on, we know that, the brain is holding memories and it's plastic. Something is changing in the brain when we go through experiences. And the candidate for all these changes are synapses. These are all the models that you learned in Reshatot. This is the Hebb rule, the famous Hebb rule that you learned about, right? So if synapses are changing, this is why it's important. And the main candidate for memory in that sense is LTP. But no one has ever shown that when you undergo memory, so it's not true. I mean, it has been shown in many, many different directions. But a clear thing to show, I'm doing a memory change, and then I see this synapse has changed due to this memory, and it's not being changed if I'm not doing that. All right? No one has shown that on that level. Okay, so it's still open question whether LTP has anything to do. I would say anything to do is probably too strong, but certainly not obvious that LTP and memory are the same thing. And it could be that it's something to do with synaptic plasticity, but LTP, this protocol where you tetanize the synapse and then it becomes stronger, maybe that's not exactly how it works in the brain. Okay. So in the next hour, which is supposed to be 2 hours, and we'll see how it goes, we're going to talk about memory and memory and spatial navigation. So let's take the break now, unless you have questions, and do that in the next hour. What? Oh, this one is not. This is going to be now. מתקשר לחברי מעבדה. כמה עולה עכבר? לקנות אותו, אני חושב, בערך 100 שקל. זה מסובך. זה עולה בערך 100 שקל לקנות עכבר בודד. זה עולה משהו כמו 25 שקל, אני חושב, להחזיק כלוב כבר חודש חיות. לשבוע. עכשיו, אם בכלוב הזה יש כמה חיות, זה לא משנה. וגם בדרך כלל הם לא מרשים לך להחזיק עכבר בודד בכלוב, אם זה לא חיה עפה. כי... בלי חלב, כאילו. חיות סוציאליות, אתה לא רוצה ש... ארוך. עכשיו זה תלוי, עכשיו יש טריק שהוא קצת מצחיק, אבל אם אתה מזמין אימא עם גורים, אז כאילו אתה משלם רק על האימא. אתה יכול להזמין גורים עד גיל של 3 שבועות. אז אם נזמין כלוב של אימא עם נניח 7 גורים, אז אתה משלם 100 שקל על הכלוב. אבל הגורים בני 3 שבועות, אז אתה צריך שהם יהיו בני 8 שבועות כדי לעשות על ניסוי כשהם בוגרים, אז אתה צריך להחזיק את הכלוב עוד 5 שבועות. אז זה 125 שקלים, 225 אתה מקבל 8 חיות במקום... זה בערך הטריק, אבל זה לא כאילו, אתה יודע... No, thanks. תדע לך שזה לא, למרות שזה לא במציאות, אף אחד לא מכיר בה. זאת אומרת, מבחינתם, מבחינת ועדת אתיקה, אין דבר כזה לימוד. למרות שהם עושים את זה בקורס שלהם, אבל מבחינתם אם אתה רוצה לדעת ללמוד איך עושים ניתוח בגולגולת של עכבר, אז אתה צריך לחכות שמישהו יסיים ניסויים, תלך לעבוד את העכבר, אז תיקח את העכבר שלו ותעשה עליו, כאילו תתאמן לפתוח לו בגולגולת. למרות שזה לא בדיוק אותו דבר, שיורד דם ולא יורד דם, יש כל מיני הבדלים, אבל כן, זה מה שהם רוצים שאתה תעשה, ואין דבר כזה עכבר שהולך לניסוי ולא יוצא ממנו דם. זה נכון, כשאיפה זה נכון. כן, מה חשבת?
שהרבה מהפירוש לא כל הקרובים מהדברים שמתאמנים עליהם זה אפשר לעשות.
יש החלטה כזאת.
על בובות.
לא על בובות, כאילו נראה לי הבובות יותר מתוחים.
לא יודע, עד 2 דקות ביום זה נראה לי שממש צריך לגלח את האן ויש לו מוגל בוילה.
כן, עם אותו דנסיטי באותו זה שאתה קודח שם ואולי מין גוש נוזל
יש דברים כאלה.
אתה יודע, כאילו עם אלקטרודה. כן, מסתבר שיש אוזני סיליקון לארנבים בשביל להזריק לשם, ויש פירושים בלי מוח שמתאמנים עליהם, כאילו בובות אחרות, בלי מוח שמתאמנים עליהם כאילו גם להזרקה, כאילו לבטן, כל מיני דברים כאלה. רק החלק של המוח עדיין לא רואה. אז כאילו בגלל זה שכאלה...
אני אגיד לך מה, כשאני התחלתי לעשות ניסויים מעלי, היו לי כל מיני מחשבות אידיאליות, אני לא פוסל את הרעיון, חד וחלילה. מעזב בחינות זה גם היה עוזר לנו דווקא לדברים אחרים, יותר לדברים אחרים לגמרי, זאת אומרת, איך למקם את המיקרוסקופ, ואיך למקם את זה כדי להגיע למצב שהוא אידיאלי, כי הכל אתה מכוון, ואז אתה שם תחבר, ואתה מגלה שזה לא מגיע לפה וזה לא מגיע לשם, אבל כאילו יש איזו נטייה, המציאות הכואבת, לשפוך לך את זה, לא שמחות שזה נגמר, המציאות הכואבת היא שרוב החיות שמומתות בכל התהליך הזה בכלל לא קשורות לזה. זאת אומרת, הרבה פעמים פשוט אתה צריך לעבוד על חיות בגיל מסוים, ואם הם עברו את הגיל, אתה לא יכול לעבוד על זה. הניסויים שאנחנו עושים, אישית במעבדה שלי למשל, ממש לא ניסויים כאילו טיפיקליים. אתה לוקח, תיקח מעבדה נחמות שלה, לא נותן בבית. מעבדה יותר טיפיקלית. שהיא מעבדה שעובדת הרבה על מולקולה ביולוגית. אתה צריך כאילו, אתה צריך על כל ניסוי כזה, לפחות 10 עכברים, לבחון את המוח, לעשות PCR או משהו בסגנון הזה, ואתה צריך שהם יהיו בגיל מסוים ושהם גדלו ושהם עברו איזשהו מבחן התנהגותי כזה או אחר וכו', ואז אם זה לא מסתדר, אז כאילו העכברים האלה הולכים להמתה, פשוט לא משתנה. זאת אומרת שבסופו של דבר רוב העכברים לדעתי שעוברים בבית חיות, הם בכלל אף אחד לא משתמש. זה אחוז מאוד קטן שבסוף מישהו משתמש. אבל אלה שמשתמשים ועושים עליהם את כל האימון הזה לצורך העניין, זה נעשה בהרדמה. זאת אומרת שזה לא שהעכבר חי וכאילו מרגיש את זה. אז כמה שאנחנו מבינים ורוצים להאמין, אנחנו לא מוכנים לו, הוא לא מודע למה שאנחנו עושים. אז אני כאילו, השאלה היא, אז כאילו איפה הרווח שלו? במה זה מתבטא? בזה שאתה מקטין את כמות העכברים שעוברים את הפרוצדורה הזאת ובסוף מומתים?
ובתקווה זה יהיה משהו גדול מספיק למעבדה כדי שזה יהיה שווה כלכלית לעשות את זה.
השאלה האמת היא, אם מישהו שמתאמן יכול ללמוד מזה משהו, כאילו אם זה שווה למישהו יוכל להתאמן על שיבוש שנראה כמו חיה, והוא יכול לראות שהוא חושד במשהו.
אני לא יודע, כי אני אף פעם לא ניסיתי את זה. אני לא יכול לדעת באיזה רמה אתה תעשה את זה. זאת אומרת, אם אתה עושה תגובה שממש היא מאוד דומה לעכבר, אז יש מוטור סקיל שאתה צריך באמת ללמוד. לדעת איך לגזור, אתה צריך לדעת איך לגזור את האור, השאלה היא באמת, כאילו לדוגמה, אתה יכול לשאול תמיד, אחת הבעיות שאני נתקל בהן, שאתה רוצה ללמוד לעשות חור בשביל להדביק הריגון בראש שלי, כי זה מה שעשינו לאחרונה, אתה צריך לעשות חור בגולגולת כדי לשים ולהדביק עליה. אז אתה מדביק את זה עם דבק טוטלי, ואז אתה מחזיק את הראש, ואז אתה צריך לעשות את זה. אם תוציא טיפה יותר מדי לחץ, אז זה פשוט נופל מהדבק הזה. ואז אתה צריך לגזור את זה מחודש ולהדביק את זה עוד פעם להציג עוד פעם. עכשיו, אם העצם בבובה הזאת היא לא מדויקת, ואם החומר שממנו יצמיע הוא אחר, כך שבעצם הדבק האנדרטלי הוא הרבה יותר יעיל, או פחות יעיל, כדי להדביק אותו, אז אתה לא תלמד מזה כלום, בגלל שאתה לא תלמד את העוצמה שאתה צריך להחזיק את הדבר הזה. עכשיו, אני מסכים שלכולם, לסטודנט, לכולם יהיה יותר נוח, לדעת שאתה מתאמן על הדבר הזה. השאלה מה הרווח, אין לי, כאילו, אין לי יכול להיות שהייתי מנסה את זה, והייתי רואה שזה, אז היינו ממשיכים,
מה שאני יכול להגיד מהניסיון הקצר שלי, שדווקא הדברים העדינים שהם כאילו לא הגזירה של האור והדברים האלה, הם הדברים שאתה ממש חייב את הדבר האמיתי לזה.
לא, אבל לא כאילו דברים שאתה חייב, ברור, כאילו בסוף יהיה לך אימון גם על בעלי חיים.
אבל האימון שאתה תרוויח, זה אימון של 3 דברים לגיטימיים בסופו של דבר.
מה אמרת שזה? לא, זה כאילו הרעיון שאם זה זדון יותר למעבדה, יותר כאילו שאתה משלם פחות כסף,
כמה זה מבחינת הכסף?
3 דברים פחות על כל בן אדם, נגיד. ואז זה נראה לי הגיוני, כאילו שברור שיש 3-4 דברים שאתה יכול ללמוד על זה.
תראה, אני אגיד לך משהו, הכסף כאן הוא לא רלוונטי. בגלל שאם זה יוכח כיעיל, אז זה סביר להניח שהוועדת אתיקה פשוט תחייב אותך לעשות את זה. אפילו אם זה עולה לך יותר. מה אכפת להם כמה זה עולה לך?
אני מקווה שזה ככה, אבל כאילו נגיד מחיפוש של כמה דקות מצאנו את הבובה הזאת בלי מוח, אבל שאתה לומד איך לעשות מדרכה בבטן. ואני בטוח שאנשים כן מתאמנים כאילו על חורים אמיתיים ולא על בובות כאלה. אז כאילו האמצעים כן קיימים.
כן, רק שהבעיה היא שכשאתה מזריק פה בעצם של בובות, לא תמיד זה אותו דבר כמו להזריק בבטן שזה...
כן.
זה כאילו... אבל נכון, בסדר, אני מסכים. טוב, אנחנו צריכים להמשיך, אבל... אתם מצאתם כבר מישהו סיני שיעזר לכם את זה?
מצאנו את מעבדת מייקרים שאולי אפשר לבוא שם.
אני יכול להדפיס את זה, יש לי מצב קצת מזיק, אני יכול להדפיס את המודל הזה.
רגע, אם אתם לא בטוחים איך העכבר יהיה בתוך הקונסטרוקציה, למה אתם לא מדפיסים מודל של עכבר?
זה שאלה טובה.
אבל שתקנו את העכבר הזה לניסויים.
כן, ראיתי עכבר שהוא בגודל המתאים.
אם יש, אז תשלחו לי, כי אני חיפשתי כזה לא מעט פעמים ולא מצאתי, ובאיזשהו שלב רציתי לעשות את זה, לעשות קאסטינג, אבל זה טיפה קצת ספורט, כאילו, זה פשוט לא היה לי לב לעשות את זה. רגע, אפילו שהוא מת, זה עדיין מרגיש לא נוח, אני לא יודע.
זה כל כך נחמד.
אני אשלח לך את זה.
מיקי, עשיתי לך רק לוודא, ממש... ממש שנייה.
כן.
רק יודע, לגבי המאמר שאני צריך להציג שבוע הבא, זה המאמר של רול מ-69.
נכון, ואל תקרא את כולו, אתה לא צריך כולו. זה יכול משהו כמו, אני אגיד לך את זה, זה עמוד.
אז אני כבר התחלתי לקרוא וכמו כתבתי את האחרון שלו.
באמת? אוקיי. טוב, אז מה, זה טוב.
גרם לי לקרוא הרבה על תורת הכבל אתמול בלילה.
אוקיי, יאללה.
אני רוצה רק לראות...
אין טעם לעבור על כל הזה.
אז תראה לי אחרי זה עד לאן אני מתחרט.
דן?
כן, אני רואה את זה, תודה.
אתה רואה את זה? איפה?
יש 7 דקות.
טוב, יאללה, בוא נתחיל. Okay, Tov, all right, so this is a bit, okay, so I'll try to do this lecture fast, but stop me if you don't, if you need me to stop. Um, so now we're going to talk about two topics which are more like system neuroscience of the hippocampus. Basically, what the hippocampus is doing. Uh, what what does the hippocampus do? So one thing is memory, and this is became very obvious after this story of this patient called HM. So now we actually can call him, because he passed away 2 years ago. We can call him by his name. Um, Anyone never heard of HM? You forgot. Okay. So HM is a famous patient that had epilepsy, and in the 60s, I think it was oh no, sorry, it was in the early 50s. He went through a surgery, and with the surgery they located the focus of this epilepsy in the hippocampus, and they basically removed the hippocampus on both sides of the brain with the surgery. And very soon after he recovered from the surgery, the epilepsy was gone, so it was successful. But gone with the epilepsy was also his. So we'll see in a second what exactly, but some kind of memory, his ability to acquire new memories. Okay, so this is what it looks in the textbook. So you can see that this is this is like a normal brain when you do a coronal section, and this is where the hippocampus is, and this is how the brain of HM looked like. And this is obviously an illustration. This is more like what really happened. And I don't know if you can see, but this is a normal brain. This is HM. There's obviously something missing here, but it wasn't only the hippocampus that was removed. So there were also part of the entorhinal cortex that were removed, and maybe others. And it wasn't exactly symmetric, as you can see from the MRI scan, that it doesn't look exactly the same on both sides. So yes. Of his brain after he's dead? I actually don't know. I'm sure they did. But I don't know if you can actually learn something from it. Because it's like, it's 40, 50 years later. So it's kind of, who knows what happened afterwards. So the process that kind of was that was was named for this thing is amnesia. Amnesia is problem with memory, and they define two types of amnesia. What is called retrograde amnesia, which means that you find it more and more difficult to remember things in the past, or anterograde amnesia, which means that it's difficult for you to acquire new memories from now on. So in terms of HM, he had like epilepsy for all these years, and then he had the surgery, and after the surgery, he basically, had anterograde amnesia, meaning that from the point of the surgery, he couldn't actually remember anything new. He was, in some ways, it's really horrible to say, but he was the ideal patient. Ideal kind of, not patient, but subject for studies, right? Because he didn't even know that he's continuing to be part of the study. What? I don't know. I mean, it's the 50s, and then you see people have done like surgeries, like taking whole parts of the brain. So I don't think they were really careful about about consent at this time. Uh, uh, so it's difficult to say. So just one word about retrograde amnesia. Retrograde amnesia, the classical example is if you go through something very traumatic, like a car accident, or you fall and you hit your head on the floor, and then you don't remember exactly what happened in the few seconds that happened before the accident, which is kind of very, very annoying. I can tell you personally from like 2 bicycle accidents that you try to really understand what really happened in the accident, but you don't remember that because you kind of these are the seconds that are critical to understand what happened are the ones that are wiped out of your memory. What? So it's not. So the idea is, I don't think it's like on purpose. I think it's more like the common understanding, and we'll talk about it in a second, is that the memory needs time to consolidate. So if you wipe the memory in the second you know it happened, if something happens, it's like, okay, let me give you the example, the example how the way we think about it. So you all know that in computers, right? I hope that you all know we have like a hard disk and we have RAM, right? You know what? Everybody knows what RAM is. So when you buy a computer, you have like, this is a CPU, this is a screen, this is how much RAM you have. So usually it goes between today it would go between 4 gigabytes to 16 gigabytes, right? And then there's a hard disk. Okay, so you work on Word, you write something, right? And someone pulls the plug out of the computer. Obviously, for all of you, you don't know that because you're working with laptops and you have battery. But like in the old days, when you had like a desktop, you would write, if the power goes off, anything you wrote before you did a save is gone, right? This is retrograde amnesia, like the proximal retrograde amnesia. Why? Because this RAM, this RAM, this RAM is a type of memory that you need to refresh all the time. So the computer takes it stores like the bits there, but it constantly refreshes them. Okay? If the power is gone, that second there's no refresh, it's wiped out. Okay? If you do a save, you save it to some kind of either a hard drive, which is actually saved magnetically, or today we have what's called SSD, which is it's like you make a flip between 0 and 1 there. It's constant. It will save there forever. Okay, so it's like a USB flash drive. This is a solid state. It's not going to change. Okay, so we believe that in the brain, in some ways, memories are stored like in a RAM for a few seconds, and then they are actually made into a long-term memory. And this takes time, and if you stop the process in the middle, then you kind of lose the memory.
Do you know the movie Memento?
Yes, exactly. So Memento is more like HM. So it's anterograde amnesia.
Yeah.
So he remembers things from the past, but every time he starts, it's like it cannot actually store the memory. So the only thing that he remembers is the things that he actually write on this Polaroid stuff, right? And this is actually everybody saw this movie? There's people. Okay, so Memento is I'm not going to tell you the story, right? But it's an amazing movie. And the idea in this movie basically is that someone like HM that cannot actually acquire new memories because of a car accident, but it's the opposite. Okay, so it has something happened. It cannot acquire new memories, and basically every time he wakes up in a completely new. He's a new person. He doesn't remember anything that happened from the day of the accident. He somehow knows that this happened, so he's using some tricks. But one of the things, the key thing in the movie is that the movie things happen one after the other, but you cannot actually make sure because it doesn't remember which thing actually happened before which. Okay, so it's actually, when you start shuffling the scenes, you can actually make different stories out of the same movie because you cannot actually tell what happened when.
Is there a lot of variance in the
Yeah, so for anterograde amnesia like HM, yeah, so it's a very famous story from the person who worked with him the most. His name was Larry Squire. I think he was mentioned here. No? I don't know if he was actually the surgeon or not. Anyway, he's a very famous person, Larry Squire, and he says that you could sit with HM for lunch, and he would order stuff and he would eat, and then the waiter will clean the table, and then he would kind of come with the menu and he will order again. Okay, he didn't remember that he had lunch. So it's about like, and then like after the third time, more or less, he kind of will feel, the waiter will ask him and he would say, I don't want to eat. But it was something like a feeling. He didn't actually remember that he was eating. And so it's something between seconds to, half an hour, something like that. That's the order of, that's the time scale. Okay, so examples, two examples, how do people assess that? So if you would give HM this test, so you give him this picture, and he needs to copy it. Okay, so this is a copy he made of the picture when he really he saw the picture and he's kind of copying it. All right. A normal person, so like if I'll take this off, after you will copy it, okay, and then I will kind of wipe it and ask you to copy it. You could do something, right? Maybe not all the fine details, but you remember that there was somewhere there was this thing that looks a little bit like a face, and there was probably this triangle. You remember some features, but if you do it like half an hour, 10 minutes later, he doesn't even remember that he made the task. He can't remember anything, right? On the other hand, when you think about this task also requires memory, right? Because you look at the page and you look at that page, you look at that, you look at that. You have to kind of maintain something in the memory in order to do that. So he could do that. So he had some kind of what we call working memory. So he can kind of hold something in the brain for a few seconds, but not for long enough. Okay. But then you give him this task. So on this task, he has to kind of take a pencil and go between the lines without crossing them, but he doesn't see, he doesn't see his. You do this trick where you do it through a mirror. So when you move, when you move right, the pen is moving left. Okay, so you have to kind of you have to learn that you have to make the opposite direction every time you move the pencil. Okay. So if you look at what happens on day one, he started with many errors, and with time he actually kind of improved a lot. Okay, this is a typical example. If you do it on day two, he started worse than he ended on day one, but much better than he started on day one. So there is some memory here between these 2 days, right? And on day three, he almost had no errors at all. Okay, what does it mean? It means that somewhere in the brain, something changed, and remembered that he has to move the hand in the other direction. Okay, but this is what we call more like a procedural memory. So it's like if you are used to use like a certain cup to drink coffee, and I'll show you the picture of the cup, and I'll ask you to hold it. You'll open the hand in the right size to hold the cup. Okay, and this is there are this classical split brain experiments on people who also had epilepsy, and they were, the surgeon would cut the corpus callosum, so the communication between the two hemispheres. So one hemisphere would know what the cup, what is a cup and what you do with it, and the other one wouldn't know what to name it, but would remember all these kind of, properties of how to hold it and what the size and stuff like that. Okay, yes.
You know if we can explicitly say that he needs to move his hand the other way?
I don't know. We can go through the like hundreds of experiments like that with him, and you can probably go and check them. I don't remember, but it's certainly what we know is that we don't believe that these specific things are stored in the hippocampus. These things that are stored more in the basal ganglia and in the cerebellum. Specifically, I think this one is in the cerebellum, but it would be one of these two rather than the hippocampus. Okay. So, so as you see, he can acquire new motor skill. He could do that. And there was a strange, observation that he moved 5 years after the surgery. He moved to a new flat, um, and he could actually draw the floor plan of of the new flat, even though it happened after the surgery. Okay, so he could kind of acquire some type of memories, and there are all kind of tricks how to do that. Probably because you walk in the room and your body actually learns the space. You can do things like that. And then, as I showed you, he had an intact working memory. So for a few seconds or minutes, he could actually remember what's going on. But after a while, it would just go away.
Does it make sense since motor skills are more like connected with that balloon and that's the height?
Again, sorry, so I don't know.
Like motor skills.
Yes.
Like more connected with cerebellum. It was spinal, right?
Yes. Yeah.
That makes some sense.
What is makes sense? That he can draw his
Acquiring
work.
new motor skills.
Yeah, exactly. But the fact that he could actually draw a map of the place he lives, this is kind of less obvious, and specifically because what we're going to talk about in the second part, which is special navigation. Okay, so without actually going into all the details, so memory, I don't know, we haven't talked about it, but memory is a fascinating thing of the brain. Obviously, it's like it's one of the most interesting properties that the brain, the cognitive process that the brain can offer, and it was studied a lot in psychology and cognitive science, and probably going. Have you done the course on the cognitive? So you probably talked about all kinds of different memories. So without getting into all the details, you know that you have long-term memory and short-term memory. In long-term memory, you can split into explicit, which means that you can actually say what you remember, and implicit, which is things like motor skills that you can show that they are there. Priming, did you talk about priming, right? So you know the process of priming. So thing like that. So we kind of we can split, and the whole point of this slide is to say that memory is not stored in one place of the brain. Okay, so it is distributed, and different kind of memory are distributed in different places of the brain. But the ability, I mean, the idea that came out of this experience of HM, and later with many repetition of this in animals, the ability to acquire new memories is happening in the hippocampus. It's not kind of exactly obvious how it happens, but the hippocampus is a key player there. So one thing to understand, just as I told you about this anterograde amnesia, is retrograde amnesia, is that memory is a process. So it's not like one thing. First of all, you have to acquire some information. Okay. Then you have to do what's called consolidation. So this is like, if you like, the save that you press in the computer. You have to kind of move this memory from this RAM to a more long-term process, and then you have to store it, and then you have to, at any time during this thing, you might be needing to retrieve the memory, okay, which is a different process altogether, okay, and actually people show that retrieval in different places of this process, different time point, can actually affect the memory. Okay, so the memory is not like something very stable, and it also for example, during this storage, it keeps transforming. Okay, you kind of you can see a picture, and then with time you would transform it into a more verbal representation of the picture, and you might remember less of the visual experience okay, but more what happened there and what did you see. So there are all kinds of processes that take time all the time during this process. But one thing that is interesting about that, I want you kind of to be acquainted with this word, which is called engram. I don't know if you have you heard it. Okay, so engram has been coined pretty early on. So here is like the citation about it. So this guy Richard Semon, influenced by a guy called Ribot, doesn't matter, but working in Berlin, was responsible for the coining of the term engram to signify a memory trace in the form of a physical change in the participating nerve cells. So the idea is that when you acquire a memory, there's some physical change happening in the brain which stores this memory. Okay? So what we think about is like either synapses are changing, or new neurons are being born, or axons are actually moving from one place to another. All kinds of things that can change in the brain could manifest this engram. But the idea is there is some physical change in the brain, and it correlates to the memory. If you want, the correlation with like computers would be changing the magnetic states on the hard drive. Okay, if you have a hard drive. Okay. Again, so the acquisition could, this is all kind of part of the cognitive course, but you know that acquisition could kind of happen in different ways. You can actually kind of incrementally be better. For example, when you do this motor skill, when you play the piano, anything that you do, when you practice more and more, you become, all the time you become a little bit better until you kind of go to some saturation where you cannot get better anymore. But there are also memories that are single trials. Something happened once and you remember it forever. Okay? And there's also this type of memories that I find the most interesting, but you know, almost no one actually works on them today, which are insight type of memories. So you kind of you try to solve a problem in reshatot, and you kind of you hit your head against the wall day after day, and suddenly, fuck, you understand it, and everything falls into place, and you understand what happened. Okay. So this is a completely different type of process.
So why is it related to memory?
What?
Why is it related to memory?
So this is not exactly memory, but you can kind of come up with also cases like that in memory, in the sense that, so it's not like, you know, it's not the inside, but it's something with memory.
It happens with retrieval, maybe, right?
With retrieval, it's a kind of a different process. It's similar, like it's similar. Yeah, you try to remember something, and you can't get it. You can't get it to work. But I think also with memory, you can kind of
Try to remember something and then
try to learn something, right? And you don't get it. You don't get it. What?
It's learning. It's not even memory.
No, but you try to memorize something.
And then you
Okay.
just remember it for good.
Yeah, maybe maybe it's not a good example. It's a good. Maybe maybe we should put it on a different slide. You're right. Okay, so it actually hits about it hits actually this point of learning, because you know it's always you often talk about memory in the context of learning, because sometimes you memorize just passively, right? But many times you remember. So actually experimentally, this is something very difficult to test. When you want to learn, you want to study memory, passive memory. So you can do that, right? You can kind of present a subject with hundreds of pictures. People have done that, right? You present a subject with hundreds or thousands of pictures, and then you know, he goes home, then he comes back a year later, you show him, you know, other pictures and these pictures, and you ask, you know, which picture he remembers or not. Okay, so you can do these kind of things. It's difficult to do a little bit with other kind of, you know, with animal experiment, but usually you try to place a memory, and you do that kind of in a learning context. Okay, so the classical context is classical conditioning. You probably all heard about that. And the point here is that not every paradigm works when you do classical conditioning, right? So if you place the conditional stimulus and the unconditional stimulus with some overlap, it would work, but in some other context, it wouldn't work, right? So I thought there was an animation up here. Does it work? Oh, okay. So this works, this works, this works, but this doesn't work. Okay. So today the most kind of the most used configuration is free conditioning, and you all heard about that in the method course, right? So you do kind of a conditioning between fear and between a painful stimulus and a sound, and you can even buy these boxes to do that, and you can do the pairing of the sound and the shock, and the animals learn that very fast, and they freeze. You know that they are freezing, right? So do you all know this study by Inbal? Anyone? No? Okay, so Inbal, before she came here, one of the studies that she made was exactly this idea of fear conditioning. So you do this fear conditioning, you couple a tone to shock, okay? But what they did is they expressed halorhodopsin. So halorhodopsin, you know, it's kind of inactivate cells when you shine light. So this is what you see down here. So you have a neuron, it's spiking, you shine the light, the spiking rate is reduced to almost zero. Then you relieve the light, and the neuron is going back to spike. And what you see here on top is that they transfected only CA1 pyramidal neurons. Okay, so they did it CA1. I don't need to explain, right? In the hippocampus, in both sides of the hippocampus, like both sides of the brain, CA1 is expressing halorhodopsin. Every time you shine a light, you stop these neurons. Okay, and what you see here, yeah, okay. So what you see, so everybody's understand what we do, right? We take a mouse, we transfect with halorhodopsin in CA1, and now we're following the following paradigm. We train the animal when the light is on. So we put the animal in the cage, and it also has this fiber optics that is on, so CA1 pyramidal neurons are shut off. They are not spiking, okay? Then you test the animal when there's no light, okay? Then you train the animal again, no light, and you test, okay? Let's just look at these four. So so this is control animal. Control animal meaning you injected a virus, but you didn't inject halorhodopsin. So you put the light on, you train the animal, you check the amount of freezing after, the day after, and they freeze about 40% of the time. Okay, you take the animals with channelrhodopsin, they don't freeze. What does it mean? You shut off CA1 pyramidal neurons, the animals are not acquiring the memory that the food shock is coupled to the tone. Okay, they don't remember, or they don't learn, or something. They don't kind of make this association because you shut off CA1 pyramidal neurons. Okay? Now you do the same thing again without the light. So the animals are becoming pretty similar to control. Okay? So now when you don't shut CA1, you get a normal response. Why there's a difference? What? Because it's always it's not a significant difference.
No, but they are after two trainings, right? The control.
Yes,
So
yes.
maybe that's the reason for the
Could be. It could be. Okay, then you wait another 24 hours. Okay, so now look here. Now you kind of you train the animal with the light off. So now the animal remembers that it knows, and you can test it, that it knows that the tone is making a shock, is coupled to a shock, okay? Now you wait 24 hours, you put it again, you test, but you put the light on, okay? So now it cannot actually retrieve the memory. So it knows it, right? But it cannot actually access the memory, okay?
But that's unexpected, right?
In what sense?
In the sense that the hippocampus is supposed to be creating new memories, but not
Storing them.
yeah.
So that I think that so what I like about this paper is up to this point. I mean, I think this is really strong. Okay, these two, right? But I think the reason Inval actually made this paper was to actually show this point that the memory is stored also in the hippocampus. And there's another part of this paper that I'm not going to talk about, which actually doing things also in prefrontal cortex at the same point. And the idea is that actually when you access the memory, you access it from the hippocampus. But if you activate, just briefly, the idea is that if you'll do the light on here, you'll do it not just when you do the test, but you'll start it about 30 minutes before. Then the animal will remember to freeze. And the idea is that then they actually retrieve the memory from another place, from the prefrontal cortex. Okay, so the idea is that you can retrieve the memory from the hippocampus, but if you cannot retrieve it from the hippocampus, it's also stored in another place, and you can retrieve it from there. All right, but if
Fulfills the redundancy?
yes, yes, but it could be also that it's stored in the hippocampus, then it moves to the prefrontal cortex, but for a long time, it takes a long time until it is being removed from the hippocampus. Okay, that's what people might think, but.
So if you do the testing, like not 24 hours later, but more days or maybe even a month later?
I think it's still there, but you have to remember that field conditioning is a very, very certain type of memory. It's like very powerful. It's very strong, so it could work differently for different type of memory. Okay, so this is kind of the holy grail of memory research. Can you actually store memories in an animal artificially? Okay, so there's a series of 3 papers. I'm presenting you the first one, but there are 2 others. Maybe one of them will happen in the presentation. This is like from I think 2 years ago, something like that, 2012, and it's using like the most sophisticated technology that you can think. So it's not only using optogenetics, but it's using molecular biology in a very sophisticated way. So did you learn about TET system in the method course? You all know? Who's done the method course? TET system. Okay. So basically, okay. So I'll just remind you. So what you do here is you put what's called TTA on a C-force promoter. Okay. C-force promoter meaning that when cells are active, C-force is it's immediate early gene. C-force is expressed. So when C-force would be kind of when C-force would be active, it will express TTA. Now you have channel rhodopsin dependent on TRE. So and TRE needs TTA to drive it. Okay, so if C-force is expressed, basically channel rhodopsin would be expressed. Except that you need this doxycycline in order to make this link. Okay, so this works only when you have when you don't have doxycycline. Okay. So what they do here is they're doing it in the dentate gyrus. You see here the dentate gyrus. Remember this shape, right? And they express these viruses only in the dentate gyrus. Okay, so now they put the animal with doxycycline in context So context A meaning like there's a cage for fear conditioning, okay, but they're not doing any fear conditioning. They just put the light on, and the animals are on doxycycline. So nothing actually happens. So this thing is break. So even if C4C is expressed, TTA is expressed, but TRE is not expressed, so neurons are not expressing channelrhodopsin. Okay. Now they wait 2 days. They take the animal off doxycycline. Okay. They put the animal in a different context. Okay. So it's a different cage for that sense. Okay. and they do fear conditioning. All right. And then they put doxycycline on again. So what happens after that? So what happened in this fear conditioning? Some neurons in the dentate gyrus, because of this conditioning between sound and shock, will be active. Okay, the idea is that dentate gyrus, somewhere in the hippocampus, we don't know where, they kind of tried dentate gyrus. Okay, so these neurons that would be active during the free conditioning will now express channelrhodopsin. Okay, and now when we put the doxycycline again, the channelrhodopsin is still there. Okay, but no new channelrhodopsin will be made. So now we put them back in context A, which is a context that they are used to and they're not afraid of. Okay, and we put the light on. Okay, so what happens when we put the light on? We activate these neurons that were actually activated during this food conditioning in B. Okay, and the idea is that what you can see here is that now when you put the light on, you get the fear. Okay, so you now you make the animal feel as if it is in the context that it was kind of getting the shock just by activating these neurons. Okay, you all with me?
Y-axis is a bit misleading here, right?
Y-axis?
Yeah.
Why? Because it's only 20%?
15.
15%? Yes, but it's kind of. But look. This is what happens before the free conditioning, right? And this is what happens when you don't have the light on. Okay. So in a way, it's kind of, it's not very strong, but it works. Okay. I'm not going to talk about the two others. There are follow-ups on that, but the idea is that you can kind of make the animal kind of retrieve the memory that it learned in a different context just by activating these neurons. Okay.
What type of neurons were they?
Dentate gyrus.
No, I mean late neurons or neurons of
I don't think they haven't. They don't have any electrophysiology together with that. They didn't record from these neurons. Okay, but place cells are usually in CA1.
You could, though, say that it's somehow a fancier conditioning, right? I mean, because you do
Fancier conditioning.
than the sound, just the sound.
Yes. Well, so we believe that when you do the sound, some neuron in the brain integrates the sound together with the shock and makes the conditioning. First of all, this is thought to happen in the amygdala, the association itself. Okay, but in some ways, yes. So you kind of activate the neurons that are kind of somehow store the memory of the cage and the sound, something like that.
maybe I didn't understand something, but isn't it more artificial retrieval than artificial?
Yes, so this is artificial retrieval of an engram. Exactly.
It's not
Okay.
artificial.
You not store the memory there. Okay, so this is the next one. Okay. So I'm not talking about it. So it's like I said, there's a series of 3 papers. This is retrieval, and then later on they actually make the engram with a very similar technique, but different. Okay. So now we're talking about the other side of the hippocampus, which is spatial navigation, and we'll do it very briefly. But this started with this technology that called tetrodes. The idea is that you take four wires, and you kind of roll them together. So you have basically four electrodes next to each other, and then when you record a spike extracellularly, you record it at the same time on four electrodes. So you can kind of differentiate between different neurons because they're coming in different special relationship to the electron. Okay, so it's like triangulation. Okay, today we can do it like in a better way by making these electrodes on a silicon piece, and then you have like different electrodes in a different arrangement. Okay, it doesn't really matter, but the idea is here you can kind of record the same spike. You see, you can see different. This is one neuron, and this would be a different neuron because the spike on this electrode is bigger, even though the spikes on the other electrode are very similar. Okay, so you can identify different neurons on the same electrode, and this is what it would look like. You can kind of connect many of them together, and this is a rat with this device on the head. And this is a first paper. So this guy O'Keefe here is a Nobel laureate for this year, basically for this experiment more or less. And what they did here is it's a funny paper to read if you want, but this is an example. You have a rat. It sits with this electrode in the hand. It sits either in an arena, and the arena has like four locations out of the arena, A, B, and C, and D. And you can see that when it sits in D, the neuron spikes from time to time, not very much. Okay, then it sits in B, the neuron is not active. It sits in A, the neuron is not active. It sits in A, again, is not active, but then it moves from A to B, and suddenly you see this burst of activity of the neuron. Then it stays in B and nothing really happens. Stays in B again. Then it moves from B to D. So it goes back from here to here. Not really a really strong thing. D to A. So it moves here, nothing happens. It stays in A, nothing happens. It moves to B to C, nothing happens. And then it goes from C to B. Suddenly you get that again. Right? And it goes on, and the idea is that in this area, when it goes in this direction, so when it goes back direction, doesn't happen. But when it goes from here to here, the neuron fires. Okay? And after they watched that happening on many cells, they realized that there's some kind of location preferences for these cells, and they named them place cells. And this kind of was followed on by many other researchers. So this is an example from the 90s by McNaughton. And what you see here is like this is the arena, and you have 80 cells, and you see that some cells fire a lot in here. So color means firing rate. Okay, so these neurons, this neuron fire here, this neuron fire here, this neuron like the corner, this neuron like the center, and this neuron like the other corner. So basically, if you just measure for many neurons in the hippocampus, you can actually tell where the rat is at any given time. Okay. Here's another example where you actually you map the arena and you find that this neuron likes to fire in this direction. Okay. Now you move the wall, you turn off the wall. So basically, as if the world has changed, and actually the cell is shifting its firing field, this place field, if you like, to the new direction. So basically, the cell cares about the relative position towards the signal that you have on the side, rather than to the. It doesn't know where the North Star. It just knows like to place himself in the arena. Some of them fire when they stay. Some of them fire when they're moving. Some of them fire when they're moving in one direction, and some don't care about the direction. There are all types of them, but mainly they they usually fire more during movement, rather than when they sit in a certain position. There are all kinds of other phenomena about it. For example, does it scale when the arena becomes bigger? So this is an example where it does, but there are examples when it doesn't. So this is kind of not exactly clear. Um, there's this interesting phenomena called remapping. So remapping is the idea that place cells are somehow coding for a specific location in a specific context. So you take a rat, here in this example, okay, and let's look at this one. I think this is better. So you look at this neuron, okay, this is a neuron, and it fires on this corner of this arena. Now you move the rat to a circular arena, okay? So this neuron that liked this corner doesn't have the corner anymore, and now this place field is in this location, here, okay? You bring it back to the previous arena, it will again, fire around the corner. You move it back to the circular arena, it fires here. Okay, you move it back to the arena, it doesn't fire at all, but you move it in this arena, it fires again in the same location. So the idea is that the same cell, just a second, so the same cell can fire have a place cells which are more or less consistent between different contexts. And kind of in one context it will code for one place, in another context it will code for another place.
Does anybody try to explain what's the connection between that corner and that area and the round space?
So no one actually says that there is, right? The idea is that it's not one cell, right? It's like you have a network, and it's kind of organized such that, when you're in this room, every neuron is responsible for some location. When you go, to the lab, same neurons are coding for different position. How is that actually working with the brain? I don't think that anyone knows. In terms of like someone has to read this information out, right? And it's ambiguous, right? The mapping is random between places, so you could also have a cell that fire here, like look at that one, right? It fires. Sorry, not this one. This one. Okay, so it fires in this direction, and on the round one it actually fires here, right? There's no connection between. I mean,
So who says that place is the thing that they are coding?
So what is our alternative?
I don't know. Could be anything else.
What? No, it cannot be anything else. If the